Rose, how's everything going?
It's good.
Are you feeling like you're going to be ready for the test tomorrow? Or not?
No, I was actually wondering if you could open up the lab again today,
I can do it as soon
but
as I'm
I
done
don't
here.
think I'm gonna be ready.
Okay. Then when you come by for, when do you think you're going to be coming by to go in the lab?
Around 1:45, because I
Okay.
have class from 12:30 to 1:35.
Okay. All right. Come by and knock on my door on your way to lab.
Okay.
All right. All right. Okay, everybody's here. Why is it not letting me? Do what I want to do. Ah, there we go. Ah. Okay. Are there any questions over the stuff we covered last time? No. Okay, well, I have questions over what we covered last time. And I think I didn't do a great job of going through parthenogenesis. So I want to go back and go over that again, just to make sure that I've made it clear what's going on. So if you were confused by parthenogenesis last time, it's probably because I did a crap job of doing parthenogenesis. So can you guys see my screen? Okay, so in normal meiosis, what happens is during the S phase of the cell cycle of those cells that are found in the ovaries and testes, you basically take the chromosomes that you have and you duplicate them so that you have a single chromosome that is now made up of two sister chromatids. And those sister chromatids of the two homologous chromosomes, they synapse, they cross over, they exchange genetic material. And when you are a female and you're creating eggs, only one of these cells that comes out of meiosis is actually going to be the cell that is available for fertilization. All of the rest of the outputs from meiosis become what we call polar bodies. And so you start off with a diploid cell and you end up with a haploid cell, in this case, an egg. And they might, in some cases, have information that is genetically not on the same chromosome as it started out because of this process of crossing over. So what happens in parthenogenic organisms is they do meiosis differently. So they have ovaries just like all the other female lizards in the world do. But what happens during the S phase is rather than just duplicating to create sister chromatids, there's an overall chromosomal duplication followed by the generation of chromatids. So you go from a diploid parent cell to a tetraploid cell after the S phase of meiosis in those gamete producing cells. And then from this point on, you can have two things happening. You can have the homologs pairing up and you can get crossing over and things like this. And once again, In this case now, you end up with a diploid egg that is available for development, whereas you had a haploid egg up here. This is actually the rarer case within parthenogenic organisms. The more common case is that the sister chromosomes actually end up pairing. So even if they do cross over, there's no mixing of the gene combinations within a chromosome, and you end up with an organism that an egg essentially that looks just like the parent. This is why we refer to these things as clones. So what makes parthenogenesis different from regular meiosis is this initial step wherein the DNA is being duplicated, the chromosomes are being duplicated. The chromosomes get duplicated in such a way that it actually not only creates sister chromatids, but also creates sister chromosomes, each chromosome with 2 sister chromatids. The vastly more common situation is that then at synapsis, the sister chromosomes pair, which then reduces genetic diversity because crossing over doesn't actually do any shuffling of the genes, and you end up with an egg that looks exactly like the genome of the mother, and it's that egg that then develops into a new offspring. And so this is how meiosis is different in parthenogenic organisms. And once again, the sister chromosome pairing route is the more common one. So up here, what they call, in this particular paper, Lutz et al. 2010, what they call canonical meiosis is what most organisms do. Premeiotic endoreplication is what parthenogenic organisms do. And this paper in particular was about Tiid lizards in the family Tiidae, which are the race runners that we have here in North America. Is that a little clearer than what I went through on Monday? Okay, good. I figured it would be. There are a new set of slides. There were also some slide duplications that got into the presentation. I've removed those slides, so there's a new uploaded set of slides that have some redundancy taken out of them that I posted on Moodle this morning. So questions about parthenogenesis? Parthenogenesis is not just restricted to lizards. I think I mentioned when we went through the mole salamanders that there are a set of mole salamanders that are up in the Great Lakes region where there's a lot of, once again, interspecific hybridization. And in those cases of interspecific hybridization, those hybrid species that come about from this hybridization, those species are also parthenogenic as well. And so when we see parthenogenesis in the amphibian reptile groups, it's almost always the result of interspecific hybridization events. And that makes actually doing mole salamander work in kind of the Great Lakes region really complicated because those mole salamanders are very difficult to tell apart morphologically because they are these interspecies hybrids. So the next weird thing about reproduction is this idea of temperature-dependent sex determination, which we find primarily in reptiles. And so once again, this is intended to just contrast what normally happens with what happens in temperature-dependent sex determination. In what we call genotypic sex determination, fertilization gives you gonad-determining genes. These gonad-determining genes are generally found on a specific chromosome. And so in humans, it's the X and Y chromosomes. And what makes you male is actually possessing the Y chromosome. Just having 2 X's makes you female. In our case, it varies from group to group as to how we classify these sex chromosomes. But those sex chromosomes contain the genes that essentially determine what type of gonad you produce. That gonad then produces hormones, and it's really those hormones that lead to sexual differentiation. So when you see sexually dimorphic characteristics in humans or lizards or whatever, it's not what makes you male or female isn't the type of gonad that you have, only in the sense that the gonads produce the hormones, and then those hormones affect your development in such a way that it turns you hormonally into a male or a female. And we'll see how this works in a minute, because I'm going to show you the results of an experiment that was used to kind of demonstrate this importance of hormones. In situations of temperature-dependent sex determination, fertilization occurs, but there are no sex chromosomes that determine sex. What happens is there is an environmental trigger, an environmental stimulus, in this case temperature, that affects a set of enzymes, hormones, and receptors that then act on gonad-determining genes, and the way in which these enzymes and hormones and receptors interact with the gonad-determining genes determines what type of gonad gets developed. That gonad formation then leads to the production of hormones, and those hormones lead to this sexual differentiation that leads to sexual dimorphism. So in this case, from here on, it's the same. What happens is what affects the formation of gonads in temperature-dependent sex determination. And what determines the formation of the gonads actually is hormone levels. And so these guys did a clever experiment. Testosterone, which is kind of the basic hormone, can basically be transformed into estradiol, which is a female-oriented hormone, or it can be converted into dihydrotestosterone, which is what leads to the formation of males. And what affects this transformation of testosterone either into estradiol or dihydrotestosterone is different enzymes. Aromatase converts testosterone to estradiol, and reductase converts testosterone into another form of testosterone. And as it turns out, temperature affects the levels of these two hormones, or these two enzymes, aromatase and reductase, in the bodies of temperature-dependent sex determination reptiles. And so they did this clever experiment where they basically varied temperature from 26 degrees to 29.2 degrees. This is in that threshold of temperatures at which sex determination shifts from 100% males to 100% females. So they started out at 26 and they went up to about 29.2, at which point we should see females dominating, whereas at 26 we should see males dominating. So when we look at this figure, if this is percent female in this figure, we see at 26 degrees centigrade and very low levels of estradiol, we see that there are hardly any females. At that low percentage of estradiol, as we increase temperature, the temperature itself is leading to increases in estradiol production, and as a result, you tend to see this shift towards females. But even at 26 degrees, if you just increase the concentration of estradiol, you take a temperature which would normally produce 100% males, and you produce 100% females because you've given those developing offspring enough estradiol that you've basically turned what should be a male into a female, given the temperature that it is at. So this is when you're adding estradiol to embryos. In this case, once again, 26 degrees is over here at 26 degrees and no aromatase inhibitor. So remember, aromatase is the enzyme that converts testosterone to estradiol. When we inhibit that aromatase by increasing the dosage of aromatase inhibitor, at no inhibition, we get 100% males at 26 degrees, and by the time we're up to 31 degrees, we have 100% females. But if you increase the dosage of aromatase inhibitor, what happens is, even at a temperature that would normally produce 100% females, you still get the production of some males, because by inhibiting aromatase, the production of estradiol at higher temperatures, which leads to more females, is reduced because you've inhibited it. And so you get higher production of males at higher temperatures because you are influencing the production, or well, you're not changing the production of aromatase in the reptiles, you just added an inhibitor that binds to it and prevents it from functioning. And so in this way, they were able to experimentally show that it's the production of these hormones that is affecting gonadal development, and then the gonadal development then affects everything else about the phenotype of the offspring. And it just so happens that the production of aromatase and reductase are basically determined by the temperature. So when we go back to this model, where temperature is affecting enzymes, the enzymes that are being affected are levels of aromatase and reductase. Levels of aromatase and reductase affect the hormones affect the gonad determining genes, that affects gonad formation, and that's how you end up with males and females. And this was just a really, really clever experiment that illustrated this. Questions about this? Now, if I'm recalling correctly, this was done with, I think, snapping turtles, if I'm recalling the paper correctly. And in this case, aromatase is produced at higher levels under warm temperatures, but in other cases, aromatase is produced at higher levels under cool temperatures. And so when we look at temperature-dependent sex determination, we'll actually see that in some species, more females are produced at cool temperatures and more males are produced at warm temperatures, which is just the opposite of this case. It just depends on how these two hormones are affected by temperature. In this model here, temperature affecting enzymes, it varies by taxa as to which enzyme is produced more at which temperatures. All right, questions about that? All right, we already did limb reduction. We did venom delivering structures. We talked about venom. I think we exhausted venom last time. Okay, then now we're to this situation here. Let me go and get something real quick. So for this round of a romp through the taxa, I'll introduce the bigger groups, and then I'll let you guys take the individual families as we go through this. So if we go, where's the phylogeny? Oh yeah, so we're going to be talking about the Geckoda first. This large monophyletic group, which represents some of the earliest members of the Squamata to have evolved. This includes all of the geckos. So when we think about the Geckota as a larger taxonomic grouping, this is one of those early reptile groups within the Squamata. Quite a few species, about 1,500 species, roughly 25% of all the lizard species. One of the characteristics of most of the families within the Geckoda are the presence of these toe pads that we've talked about before. They're generally nocturnal, although there are groups of day geckos. I'm thinking primarily of the day geckos that are from the island of Madagascar. Once again, they are mainly oviparous. They lay a parchment shell. For the most part, they lay parchment shells in certain families, but everybody else lays calcareous shells. So the geckos that I'll collect in March and bring back that we'll play around with a little bit in class are ones that form calcareous shells. So calcareous shells is the norm, parchment shells is the exception. Some species are parthenogenic. So the parthenogenesis model that we just looked at before would apply in those taxa that are parthenoform. And then there is also temperature-dependent sex determination in a number of different groups within the Geckoda. Tail autotomy is super duper common, and also skin loss as a defensive mechanism is also super common. What will happen in skin loss is, let me go to this figure, is when you capture a gecko and you have it in your grasp, what it will do is it will twist its body, and it will twist its body, and as it does that, it will shed all of the epidermal layers of the skin, and it will basically just squirt out of your hand as this naked lizard, and you're left with a bunch of scales in your hand. And so that's one way in which these lizards avoid predation, is by shedding their skin and then just sliding out of their skin, so to speak. This individual here has lost a big chunk of its scales as it was captured, and this individual has also, in the past, lost its tail. Remember that when you regenerate a tail in the reptiles, it appears as a slightly different morphology in terms of the scalation of it. So when I talk about skin loss being common in this group, that's what I refer to. This happens more easily in some groups than it does in others. The Madagascar day gecko that I have at home as a pet is very prone to losing skin. It lost big patches of skin when I captured it in the field in Florida. But the Mediterranean geckos, they do lose skin, but you have to handle them pretty roughly for them to lose skin. So skin loss as a mechanism varies quite a lot within the group in terms of its efficacy. These are the toe pads that I talked about before. This is a typical gecko toe. Each of these lamellae are made up of rows of really ultra-fine brushes that are extensions, once again, of the Oberhauten level of the skin, that outermost layer of the epidermis. And when you look at these, so these are all scanning electron micrographs. When you look at these, they look like little bundles of broccoli, but when you blow them up really, really high levels of magnification, you see that they're these little spatula-shaped structures. And when you look at how they interact with the surface, what you see is this structure basically bonds with the surface. And this is all done with van der Waals forces. So I don't know if you guys remember about all of your intermolecular forces from chemistry, but van der Waals forces are these really weak intramolecular forces. They're not as strong as hydrogen bonds, but they do provide some attractive force. And it turns out van der Waals forces are responsible for taking each of these little spatula shapes and adhering them to the surface. And what enables a gecko to support its entire body weight via these toe lamellae is that each of these little clusters of broccoli-like setae have on the surface of them hundreds or thousands of these little spatulas, hundreds or thousands of those little spatulas on each of these little blocks, on each of these little tips of the literally hundreds or thousands that occur on the individual foot of a gecko. All of those combined van der Waals forces, which are very weak intramolecular forces, those combine together to be able to support the weight of the lizard. This is Mediterranean gecko. We'll see these in class, in lab when I get some from the field. And you can see here, this is the toe of the gecko, and this white structure underneath, this is the toe lamellae, the toe pads of the gecko that you can see just poking out from the edge. And as a result, it can adhere to rough surfaces really easily, like this brick that this one is on, but they can also, these guys can locomote across glass out in the field. I'm gonna get out of this for a minute. There is actually a group. Can you guys now see my web browser? Okay. Whoa, Need to get out. Why am I in Moodle? Something is definitely wrong. Oh. So there is a group at UMass Amherst, which is working on applications of Gecko topad structures to basically create a skin. Let me see. I haven't been to this website in a while. Yeah, Duncan Urshik is part of it. So they're actually using this. Can you guys hear this? Can you guys hear the audio? Okay. So anyway, I'll just show this video and I'll kind of narrate it for you. But basically, they're creating removable adhesives. So they just put a gecko skin structure on that door and hung a computer monitor from it, and it just pulled right off, just like a gecko's toe would pull off something. Here they're going to put it on a painted wall. They're going to adhere it to the painted wall. It's going to support the weight of that computer monitor. And when they pull it off, it doesn't do any damage to the wall because it's just adhering via these van der Waals forces. Now notice that they use this applicator to push it upwards, and when they pull it off from an edge. And when you pull it off from an edge, what it's doing basically is it's essentially breaking each of those van der Waals forces one at a time. Whereas when you pull down on all of them all at once, the combined force of it holds that thing in place. So you apply it, and as long as you're pulling it unidirectionally, all of those van der Waals forces are in contact with the surface. But then when you pull it off, you pull it from the edge, and it just comes right off. So they're developing this for doing things like hanging a computer monitor. They're also developing these things for like police forces and special forces and things like this to enable people to easily scale walls in those types of environments where you might want to scale a wall quickly and easily. And there's no power or anything to this. It's all just mechanical forces at play. So that's pretty cool application of Gecko technology. All right, so let's get back. Are you guys back to my my PowerPoint? Okay, so Quinn, you had the spherodactylity, right?
Yeah.
All right, tell us about the spherodactylity. I've got some things up here, but you might add some things. And for that matter, I might add some things.
Okay. So I had written down most of the stuff he has on this slide, but I also, there's about 200 species and 12 genera. There's only one species that naturally occurs here. They're also found in Central and South America and the Caribbean.
Who's that one species?
I do not know.
Okay.
They're found in the Caribbean, Europe, and South Europe, the Middle East, and Central Asia. And then one of the species, the gonatodes, they lack topads. And then there's another species, the Sphaerodactylus. They do have topads, but they're small round pads, and they have on a large scale on the tips of their toes.
Yeah, so in your in your field guide on plate 24, these are species of Spherodactylus lizards, Spherodactylids. One of these is Gonatodes, the others are in the genus Spherodactylus, and they are, for the most part, all introduced except for one of them that is native to Florida. But yeah, they are more common throughout the Caribbean. I've seen them in Puerto Rico before. They live mainly down in leaf litter, so they're not a climbing gecko, which also explains why they don't have well-developed toe pads. Did you mention the egg?
No, I didn't talk about it. I just said I had
Okay.
it written down. If you had
Right. Yeah, so you'll see that geckos will either lay 1 or 2 eggs typically, and it just varies by group as to which it will be. One of the cool things about geckos is they have kind of a translucent skin in many cases, so you can actually see the eggs developing inside of them. Anything else you want to add, Quinn? All right, so that's a Spherodactylity. So here's what they look like, and this is their distribution. Once again, they get into extreme South Florida as well, and then there's some introduced species in the United States. The Geckonidae, these are what we call the spectacled geckos, and they are they are called spectacled geckos because they have a scale that covers the eye, and it's transparent, and they see through it just like a snake would see through it. So they don't have eyelids. So this is one way to distinguish them from other types of geckos. All of these species are, for the most part, either arboreal or what we call scansorial. Scansorial is spelled S-C-A-N-S-O-R-I-A-L. Scansorial geckos, or any organism that is scansorial, is a species that tends to crawl around on rocky surfaces, vertical rocky surfaces. It's a little weird that we would have a term that is that specific, but we do. Because they are accustomed to climbing around on these vertical rocky surfaces, cliffs and things like this, this is also the group that tends to also form commensal relationships with humans. And it's because they do a really good job of crawling around on human habitations that are made out of brick and masonry and things like this, stucco. And so when they see a warehouse or a human house, they just view it as one more cliff to climb, and they oftentimes will be the types of geckos that get introduced around the world. The Mediterranean gecko, which we'll look at in lab, is a member of this family. These guys sometimes have communal nests such that a number of different females will lay eggs all together in the same little area, same little egg laying place. They're not terribly communal in terms of their behaviors. They can be quite aggressive towards one another, but they will lay eggs in one spot, probably not because of any social behavior, but simply because those spots have the right climate to incubate the eggs well. And then not all of these, but a number of them are also parthenogenic. So this is where we see parthenogenesis reasonably commonly. Questions about the family Geckonidae? All right. And this is their distribution. They have a really wide distribution. In the United States, they're mainly found in the southern parts of the United States. Eublephaриdae. This would be Kiara.
Yep. So like he has on there, they're known as the eyelid geckos. And they're known for that because they're the only geckos that retain functional eyelids. So in others, they're covered by an immovable spectacle. And they are also nocturnal and inhabit inhabit terrestrial environments. And they also lack topads and subdigital setae that give the geckos good climbing ability. And for habitat, they occupy arid or subhumid environments. And then they occur in Western and Southern North America, including California and Texas, and through Mexico on the distribution, and then some portions of Africa and Asia.
And you'll notice that oftentimes these places where they occur are in drier areas of continents. And so in Texas, we have the Texas banded gecko and the Big Bend gecko in the genus Coleonyx. There are also other members of the genus Coleonyx out in the desert southwest of the United States. And so this actually is a leopard gecko, which you can find commonly in the pet trade. It's from Africa originally. These make good pets because all of these species are on the less aggressive side of geckos. The geckonids are oftentimes considerably more aggressive when you encounter them in the wild, and some of these can get actually quite large. The tokay gecko is a very large gecko that actually has a fairly painful bite from Malaysia. What type of eggs do they lay?
I don't know that.
Okay, so the eublephaриds, if I'm recalling correctly, lay a parchment shelled eggs, just like the, they lay parchment shelled eggs among the geckos. And when we look at them in the phylogeny of the geckos, let's go back to that. You will see that they're once again, among some of the earlier groups to have broken off and evolved separately. So they're oftentimes viewed as the more primitive of the gecko species. All right, so now we have Skinkomorpha. So we have another monophyletic group, and the Skinkomorpha includes the skinks and the Xanthuciids. Skinks we have commonly in the United States, and Xanthuciids we have commonly in the United States. Chordilids are, once again, old world groups. Once again, a really species-rich group, all kinds of different body forms, everything from reasonably stout-bodied, stout-legged organisms to longer, more slender species that have reduced legs or reduced digits on the feet of the legs, all the way down to legless varieties. What makes all of these, what kind of unites all of the members of the Skinkity. Oh wait, I'm doing the Skinkity. Oh, Elizabeth was supposed to do the Skinkity. I went to Skinkomorpha and then I went to Skinkity. Sorry, Elizabeth, I stole your thunder. Go for it.
No, it's okay. So I had that down, and then I said that most of them are litter or sand swimmers. So they kind of burrow into the loose soil and move through it that way. Mm-hmm. Most are diurnal, but some can be nocturnal or active at twilight. They're viviparous with a complex placenta that can transfer proteins and lipids. They have the cycloid scales, which are smooth and hard, and they have smooth edges. And the osteoderm underneath is composed of multiple small bones fitted together rather than one large bone beneath each scale. And then one subfamily has a secondary palate that separates the mouth from the nasal cavity. Limb reduction has evolved more in this family than any other lizard family. And they have tail autonomy and can regenerate their tail. And then they're found in North America, South America, Africa, Europe, Asia, the oceanic islands. So pretty much everywhere except for where it's too cold or desert area.
Although they're commonly found in the deserts of Australia. So Taliqua, this is the blue-tongued skink from Australia. This is found actually in the desert regions of Australia. one of the things that you'll notice when you see these in the lab is the first thing that you'll notice is the scales, and the scales of skinks are very distinctive, and it's hard to mistake a skink for anything else. Elizabeth mentioned this sand swimming sort of morphology. That's what this species is here. It's one of the legless species that occurs in sandy habitats. But even in leaf litter, when you see these things running, you'll see that most of the times they run with a kind of serpentine motion. And so once again, even if you just catch a flash of a skink running through leaf litter, it's easy to identify it as a skink because of the way it runs. Elizabeth, what's going on with the tail of this guy here in figure D?
I'm not certain. I don't know if it's like a mating behavior. I don't know.
It's not a mating behavior. What you see oftentimes in skinks is when they're very young, they will oftentimes have a blue tail, and it's almost always blue, this iridescent blue. And as they grow, they'll shed a new set of scales that then lack that iridescent blue. And it's believed that when they're younger and smaller and they're more prone to predation, the blue tail attracts attention to the tail, which then can be autotomized and allow the animal to get away, and then they lose that coloration as they get larger and older. So if you see a 5-line skink here in Missouri and it has a blue tail, that's a sign that that's a juvenile individual. And some people refer to skinks here in the central and southeastern United States as blue-tailed skinks, but there are a number of species that have blue tails. That's not a good distinguishing characteristic. All that tells you about the animal is that it's immature. All right, Rose, tell us about the night lizards.
Okay, so they live in the United States, Cuba, Mexico, as well as Central America. And people rarely see the night lizards during the daytime because they can be active at both night and day if the daytime temperatures aren't too hot. And females of the night lizard species, except one, give birth to baby lizards, and then one of them lays eggs. The Cuban night lizard lays actual eggs. And then, like you said about the species, they don't have working eyelids, so they see through a scale covering the eye called a spectacle. Yeah.
So if you're in the desert Southwest, like if you're out in why am I losing the name? Joshua Tree National Monument. These Joshua trees are basically big yucca plants, and they shed their leaves, and at the bottom, at the base of these yucca plants, you'll see this big pile of old leaves that don't decompose very quickly because it's a dry desert environment, and so things don't decompose very quickly because it's not very humid. And if you dig around in these piles of old dead yucca-like leaves at the base of Joshua trees, you'll oftentimes find that this is where night lizards in the desert Southwest occur. So anywhere that you see a buildup of leaf litter around vegetation in deserts, you'll oftentimes, if you start digging through those things, you'll find night lizards. You don't see them, people don't see them very often because they don't come out during the daytime. You actually have to know where to go looking for them to find them during the daytime. The Lacertoidea, this is the next big group. It includes a number of different families. The Teiidae are ones that occur here in the United States. And then the Amphisbaenids also have limited representation in the United States. All the rest of these are for the most part not found here. So we have one representative, the Rhinuridae, once again, that occurs in Florida. But this large grouping, the Lacertoidea, this big monophyletic group, is mainly represented by Tiids in the United States. And the problem with this group is that there are no morphological synapomorphies. The monophyly of them is supported primarily through molecular evidence. So this whole phylogeny that we operate off of, this is a molecular phylogeny that has been relatively well supported over time. So Kelsey has the Tiids.
Yep. So this is made up of the whiptails, race runners, and tegus. They're native to the Americas. So the next slide has the map that shows most of the US and then South America. There's 150 species in 18 genera, and it includes several parthenogenic species. They're diurnal, they're very active, and they're found in arid to humid habitats. And then they also have large rectangular scales that form like transverse rows ventrally, and then they have smaller granular scales dorsally. And they also have a forked snake-like tongue. And their head scales are separate from their skull bones.
Yeah, so one of the things that you'll notice in some of the descriptions of the families, you'll see that the scales can either kind of conform to the outline of the bones that are underneath, or they can be kind of divorced from that, such that the scales, the structure of the scales on the head don't really reflect the bones that are underlying. And in species where there's heavy scalation on the head and face, oftentimes you'll see that the scales actually correspond to the underlying bones, but it doesn't in this case. So you can see in this photo, you can see those belly scales that are rectangular, and it's a little harder to see on this specimen, but easier to see on this specimen. This specimen looks almost smooth, but it's because the scales are what they call granular scales. They're just little bumpy scales. They don't overlap one another at all. We have a representative of this family up in the lab that you'll look at when we start working on the reptiles, and you'll be able to see those granular scales really easily when you put it under a dissecting scope, which is something that you probably should do. Kelsey mentioned the forked tongue. Oftentimes when species have a forked tongue, it's a sign that they're using that tongue for detecting scent. And you will oftentimes see these guys tongue flicking while they're out foraging. The species that occur in the desert Southwest are in the genus Aspidoscelis for the most part. And those guys are oftentimes prey specialists, and they particularly in many cases like preying on things like scorpions. And they actually have very specific foraging behaviors related to capturing and killing and eating scorpions. And they will oftentimes go along, and their pointed snout here, they will actually put that underneath rocks, and they'll flip those rocks up, and underneath those rocks they'll find scorpions in many cases. And so they tend towards a dietary specialization in the desert Southwest of the United States. Questions about Tiids? Kelsey, you mentioned parthenogenesis. What results in parthenogenesis? What do these species tend to do?
Wait, what?
So parthenogenesis is relatively common in this family. What is that arising from?
Oh, I don't know.
This interspecific hybridization tends to be more common in this group than some other groups. So all the groups in this family that are parthenogenic are either known or suspected to be hybrid species that are from interspecific hybridization events. All right, Quinn drew the amphisbaenids, which are a very unusual group of lizards. They look funky. They're elongate. They're mostly legless. Some of them have 2 front legs. The right lung is reduced. In these long serpentine species, you will oftentimes find that in species that have elongate bodies, they have reduced the lung. And so rather than having 2 lungs, they will have 1 lung that is just really long, and the other lung will have disappeared over evolutionary time. The brain is completely encased. They locomote using concertina motion, which is the kind of locomotion that earthworms use, where they basically compress their body and kind of squeeze the front of the body out in front of them, and then they anchor that body, and then they'll pull the rest of the body up behind them. And we'll study that in more detail. Certain snake species also use that form of locomotion as well. And they have these prominent annuli. So these are characteristics for the entire group Amphisbaenia, this entire monophyletic clade. But then, so this is the Amphisbaenia within the Lacertoidea. And then Quinn will tell us about the Rhinuridae.
So there's currently only 1 living species that's only found in Florida. They have no legs. Their eyes are covered by scales, and then they have no external ear openings. And so,
Yeah, and this is what it looks like. It's this long pinkish snake found in sandy areas in central Florida. Were you guys around when Dr. Schaefer was still here? No, she had already left. Yeah, okay. Dr. Schaefer worked in this central Florida, and there are these kind of sand hills, these not very tall sand hills that occur in central Florida, and that's the habitat of this particular species. And they look more like snakes. People, when they do find them, will mistake them for snakes because of their lack of limbs. But most people don't ever see them because they are largely burrowing. Bipedidae, which is this group here, is a type of Amphisbaenia that lives down in Mexico. And it's one of the groups that has 4 legs and lacking hind legs. But the other groups are all limbless for the most part. So the Anguimorpha is the next big group, because we're skipping Serpentes because we'll cover Serpentes all by themselves. Within the Anguimorpha, we need to be mainly concerned with the Helodermatidae and the Anguidae here in the United States, but we're going to talk about the Varanidae also, because they're just cool lizards, interesting lizards. So who has the Helodermatidae? Oh, I have the Helodermatidae. So the Helodermatids include the Gila monster and the Mexican beaded lizards. At one time, these were kind of all lumped together into one wide-ranging species, and they've since been divided up into a number of different species. These are relatively large, relatively thick-bodied animals. They have a prehensile tail that is used to store fat. And from that fat, they can metabolize that fat, and it serves as a source of water for them. They are venomous, but in this case, the venom gland is not muscularized. And this is a venom gland that is derived from a salivary gland in the mandible, in the lower jaw. And the teeth of the Helodermatids are actually modified with a little channel that kind of acts like a capillary tube for transmitting the venom that basically is released out of the salivary gland into a groove of the jaw, and then it's picked up by the teeth and transferred. These guys are oviparous, and they feed on a wide variety of vertebrate prey. They'll basically feed on almost anything that they can find. And they are found primarily in the desert Southwest and then down into Mexico. And into Mexico, they can then be found in more moist regions of Mexico and Central America. So this is the head of the Helodromatid. The venom gland is very large. And once again, these teeth, this is the channel that the venom is released into. And then they have these grooves on the front of the teeth that kind of act like capillary tubes and draw the venom up. And each of these teeth have these groove-like structures in them. So unlike the snakes, where there will be a single pair of fangs or sometimes two pairs of fangs, all of the teeth of these Helodromatids have this grooved envenomating capability. But unlike the snake venoms and snake fangs, in that case, the salivary gland is actually connected to the tooth, and the tooth is hollowed out, and the duct from the salivary gland goes into the tooth, and that's how the venom gets to whatever the prey item is actually by sending it through the interior of the tooth, as opposed to just a surface structure. This is the Helodromatids, and this is the one that occurs in the desert Southwest of the United States. The ones that occur in Central and South, in Mexico and down into Central America are gonna not have this red coloration. They're either gonna be solid black or black and white reticulations and spots. Who has the anguity?
I do.
Kiara.
So they're known as the slow worms, glass lizards, and alligator lizards. They're widespread, primarily in the tropical and temperate regions. Most are terrestrial, but some are burrows and arboreal. Their habitats include open grasslands, sand dunes, pine oak and cloud forests, lowland rainforests. So yeah, a lot of habitats. Most are diurnal and prefer cool, humid environments. And specifically for the glass lizards, they have movable eyelids and external ear openings. And the scales are reinforced by osteoderms, which we talked about earlier. The tail autonomizes readily, and there's limb reduction and loss occur and has evolved several times. And then one of the species, I may be pronouncing this wrong, Ophioids, Ophioids, has lost four limbs but retains tiny hind limbs. And several of the genera are viviparous, but others are oviparous. And then numerous species attend their eggs during incubation.
So this is actually the European glass lizard. You'll see that it has a very lizard-looking head. You'll also see that it has this fold that goes down the side of the body, which is common in both the Anguini and the Geronodidae. These are the alligator lizards that occur in the montane regions of the West. This is a glass lizard that occurs in Europe, and there's also a species of glass lizard that occurs in the southeastern United States. Are any of you fans of Indiana Jones movies? Have you seen the Indiana Jones movies?
I saw them a long time ago.
A long time ago, okay. Indiana Jones, Raiders of the Lost Ark. Do you recall a scene in the Indiana Jones movies, in the Raiders of the Lost Ark, that would be of interest to a herpetologist?
The scene where they had all the snakes on the ground, and it was like a bunch of snakes that should not be together, just in like
Okay.
a hole in the ground.
So among the things that were wrong with that is the snakes that didn't belong. But the other thing that was wrong with that is the snakes that weren't actually snakes. So I don't think you're going to be able to hear the audio on this, but you don't really need to. Can you see YouTube? Okay. I'm going to make this full screen so that we can actually look at this. And we'll finish with this today. So here they are. They're being lowered into this tomb with a ton of snakes. Snakes. Indiana Jones is going down. Okay, first of all, this is a boa constrictor, which is native to South America, so there is no reason at all for it to be in Northern Africa. So our next topic after we finish going through the reptiles, we're going to be talking about biogeography. And once you learn a little bit about biogeography, you can't watch movies that are set in the earth any longer because you see stuff like this. But one of the things that you'll also notice here you can see the body of a snake-like organism. But one of the things that you'll notice right here is you will see this lateral fold that goes all the way down the body. This is actually a European glass lizard. And we'll just consider, there's another glass lizard right here. This is an actual snake. I'm not sure what species that is. And we'll continue watching this terrible movie making. Look at these snake-like things here. You see that there are these plate-like scales that go across the back. You can see that in the reflection off of the skin right here. This is basically just a gigantic pile of European glass lizards. These are all anguids. Once again, this is a Burmese python, which is native to Southeast Asia. It is not native to Africa. And you will also notice that it is slithering across glass lizard, 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 glass lizard. These are all glass lizards. Continuing, this is a reticulated python, also from Southeast Asia. And once again, you can see that here is a glass lizard. It has a very lizard-like head rather than a snake-like head. And you can also see that lateral fold going down its body. This is actually a cobra. And if you rent a DVD, if you pay close attention and cycle through this, you can actually see at one point there's a reflection off of the plate of glass that is separating Harrison Ford from the cobra, because this is actually an actual cobra. And a lapid snake, which we'll talk about when we get to the snakes. Oh, come on, get back to the glass lizards. Okay, so here's a cobra, and here's a glass lizard right in front of the cobra. And I'm not sure what species of cobra that is, actually, as it turns out. Once again, back to that same reticulated python. So what happened was they needed this scene to be full of snakes, and they kind of exhausted all of the snake suppliers who supply snakes to things like film sets and things like this, and it wasn't nearly enough to fill up the set. And one of the things about the European glass lizards is they tend to aggregate at certain times of the year, and so you can collect them when they're in their hibernacula. And so they sent out a notice to everybody who might be interested in this kind of thing in Europe, and they gathered up all of these European glass lizards that basically stood in for all of these snakes in this particular movie. So snakes in movies can be quite infuriating because film directors don't know anything about the biogeography or the morphology of reptiles. They just need something that will scare the crap out of audiences. So we'll end there with some nice footage of a bunch of glass lizards in a tomb in Egypt. So, questions? All right, tomorrow we will be taking the first lab quiz, and I hope to have that done by 10:45. If I'm running a little behind, don't be all that surprised. It takes some time to set that up. Our other plan for tomorrow was after the test, we would begin working on the reptiles. But as it turns out, there are a great many more reptile species than amphibian species that we have in the collection. And it turns out that Dr. Shi needs the room on Friday for an anatomy exam. And so rather than bring all of the reptiles out only to then have to put them all back away again, we'll just end lab after the exam tomorrow. We'll put away all of the amphibians, and then the lab will be relatively clean so that Dr. Shi can run his lab exam. Because of COVID situation and keeping spacing down to a minimum, he's going to have to use 2 labs to run his lab exam rather than 1. So use that extra time that you have in lab to continue to work on your dichotomous key, which will be due next week in lab. And then we'll start going through the reptiles next week in lab rather than this week in lab. All right, questions? All right, I'm going to go unlock the lab right now, and it'll be open for the rest of today. And I will see you guys tomorrow at 10:45. Rose, do you want to come